Hello and welcome everyone to episode 81. Today I'm going to take you on a journey exploring the immune system in animal organisms. Now this is going to be a really exciting and unique episode because it's going to be a fair bit different from a lot of the others. Now in previous episodes I've talked about how animals interact with physical stuff. In episode 74 I talked about how animals use water and how water is regulated within their bodies. In episode 76, I talked about how animals sense inputs from their environment, including a small range of detectable sound and light frequencies, temperature, and even electromagnetic fields. In episode 79, I talked about the physical constraints on biology that dictate form and function. And in episode 78, I talked about animal motion and the physics of manipulating a body so that it moves through the world. And in the most recent episode, I explored gas exchange and how physical gases like molecular oxygen and carbon dioxide are breathed in and out by animals. Now, all of these things are about animals interacting with non-living things, with non-living matter, with physical matter in their environment. Only episode 75, Animal Nutrition, did I explore how animals interact with other organic elements in their environment, like herbivores or carnivores eating their respective prey organisms. Analogous to the third episode of the previous season, today's episode will also touch on a biological variable in the animal's environment. In any discussion about the immune system, there has to be a discussion of what the animal needs immunity from. And in a very general sense, the animal needs to defend itself from pathogens, or harmful microbes or particles in the environment. Now, I've often described animals as massive chemical superstructures, because not only do I think that's accurate, but I also think it puts life in a very interesting perspective. Animal cells, and all other cells really, they're all ordered conglomerations of chemicals, interacting in chaotic but regulated, genetically determined ways. In multicellular organisms, like animals, many dozens to trillions of cells compose a larger, massive, singular being that has some degree of awareness of the world around it that transcends the individual capacities and awareness of any individual cell. Now, from this angle, the animal body is an amazing orchestra of chemical reactions, moving elements and nutrients and ions throughout this body structure in pulsing waves as food is digested and absorbed. The chemical superstructure is defined by this inward to outward flow of energy and chemicals, as nutrients are absorbed from the food tube running through the core of the structure and distributed through the vasculature and across the cellular architecture of the entire body to sustain the entire thing, to sustain this dynamic structure, this form through time. Now, on the cellular level, the driest, deadest, outermost layers of the animal superstructure are constantly flaking off, only to be replaced by new cells being pushed up from the basal membranes deeper in the dermal tissue. Now, I'm being somewhat poetic in these descriptions, but I'm trying to give you a particular view of the animal body, so that you might better understand what happens when a pathogen is able to sneak inside and interfere with it all and ruin everything that's going on. Now, the pathogen is a very tiny chemical superstructure that enters the animal body through wounds, or orifices, or body fluids, or membranes of some kind. These pathogens can be small parasitic worms like helminths and tapeworms, and they can be fungal spores that infect and grow out their mycelium within the animal's body. Bacteria, amoeba, and other single-celled microbes are other forms that pathogens can take, as are the barely counts as living, probably not living, molecular complexes called viruses. All of these pathogens, be they multicellular helminths, single-celled bacteria, or tiny viral particles, they can all cause infection and disease should they get past the animal's biochemical defenses. These biochemical defenses are known as the immune system, which works both passively and actively to keep pathogens out and to maintain the health of the animal. An animal will have multiple defenses, including innate defenses and adaptive defenses. The innate immune system occurs in all animals across the animal kingdom in many forms. Now, while the innate immune system has a rapid response to the pathogen, the innate immune system is also indiscriminate. It's a generalized body response to the presence of a pathogen. The innate defenses cannot detect specific pathogens and make specific, focused, tailored responses. It has no memory of previous infections. Those traits, having a memory, being able to make specific responses, these are all qualities of the adaptive immune system. The adaptive immune system is slower to respond, but it's much more targeted, it's much more precise, and it retains a memory of previous infections, so that when a similar pathogen is encountered, the response is faster and stronger and even more targeted. In the context of human civilization, this is the whole idea behind vaccinations. Vaccinations are like training for the immune system, and as a medicine, they've had world-changing benefits. Anyways, the innate and the adaptive immune systems operate to maintain the health of the animal, to maintain the proper ordered functioning of this giant biochemical superstructure. But, as we all know, it's not perfect. Disease is a terrible and prolific dealer of suffering and death, and despite the animal's best biochemical defenses, some pathogens are just too clever or too dangerous. Through whatever mechanism, they can destabilize the animal's chemical superstructure, doing lethal damage, or creating problems that induce debilitating injury, or susceptibility to secondary infections, all of which eventually can lead to death. Thus, there is very strong evolutionary pressure on behalf of the animal to develop sufficient immunity to the various microbes and microorganisms that saturate its environment that they have to evolve some of these immunological defenses. Otherwise, their bodies would just be too susceptible. Their lineage would be taken out too quickly. There's no escaping the microbes and the microorganisms. They exist on everything. You can't keep them out, you can't avoid them, so they must be dealt with on a physical and a chemical level. The first line of defense against these microbes in virtually all animals is innate immunity, and the first defensive structures of the innate immune system are the skin and the mucous membranes. The skin, be it leathery mammalian skin, the scales of a fish or a reptile, or the porous skin of a frog, form a physical barrier between the animal's internal body, its organs and nerves and gonads, and the outside environment. I probably shouldn't have mentioned a frog, because amphibians, with their more amphibious skin, this is less true for them. Their skin is more porous, and so they are particularly susceptible to, for example, environmental pollutants, as well as other pathogens. So I guess this is a good counter-example. Now, in many cases, the animal's skin is waterproof. It's sealed, and it provides a complete barrier. The insects, in particular, can also coat their bodies in a waxy cuticle layer, much like the plants do on their leaves, to make themselves waterproof. And for both the plants and the insects, this is a quite effective mechanism for keeping out pathogens. Where the animal has a natural orifice, like a nostril, an eye, or a mouth, there's some kind of mucus or fluid secretion that lubricates and protects the soft membranous tissues. So for example, in the tissue fluid around a mammal eye, there's an enzyme called lysozyme, and it works as a general antibiotic. The fluid, or the mucus, can entrap pathogens, and it makes them easy targets for other protective enzymes, or for immune cells, which I'll get to in a few minutes. Now, the pathogens can overcome this defense with their own enzymes that allow them to chemically burn, burrow, or bore through the skin, or disrupt the mucus lining of the nose or throat or eyes or whatever. Sometimes pathogens exploit an open wound to gain entry into the body, while other pathogens have evolved to live around the animal's genitals, so as to be spread to a new individual when the animal has sex. And these genitals are lubricated. If there's any orifices, they're often opened up. There's mechanical stimulation, mechanical exchange of fluids. This is an environment where it's really easy for pathogens on one animal to be spread to another. However it happens, in any situation, when the pathogen gets past the skin and the mucus and penetrates into the animal's body, the innate immune system's chemical defenses come online. So at this point, this is the beginning of the true innate immune response, which, as I said, is a generalized response that does not develop a memory of pathogens. First and foremost, the white blood cells, or the leukocytes, will gather in the blood to the point of infection. They'll seek out and try to hunt down the pathogenic microbes and destroy them. This is a generalized response, which means that the innate immune system, it can differentiate between a bacteria, a fungus, and a worm, these major divisions between pathogens, but it can't detect any specific genera or species, and it can't mount a specific response to a specific pathogen. The innate immune response is only able to determine the broad general category of pathogen thanks to a class of molecules called pattern recognition receptors. These pattern recognition receptors detect particular types of antigens produced by each type of pathogen. So an antigen is any molecular residue that's given off by the pathogen, like a secretion from a bacteria, or some kind of tissue marker from a parasitic worm. These pattern recognition receptors are a broad class of receptor enzymes that detect a similarly broad range of antigens, and through this mechanism they allow the animal to identify roughly the general type of antigen, whether it's like a bacteria, a fungi, a worm, etc. Some of the molecules that these pattern recognition receptors can bind to include characteristic lipopolysaccharides on gram-negative bacteria, or single strands of viral DNA, or a fungal molecule called zymosan. So these pattern recognition receptors, it's a decent little toolkit for all of your generalized immunity needs. But it should be clarified that these pattern recognition receptors are present in both plants and fungus, which implies that they're a basal quality of virtually all eukaryotes. So from an evolutionary perspective, the receptors that detect pathogen antigens are extremely important, and they've been preserved across an explosively diverse branch of the tree of life for around 500 million years at least. All animals have these pattern recognition proteins as well. They all have them expressed on the surface of their white blood cells, and when a particular receptor binds to its target antigen, it initiates a specific response. This response typically involves the white blood cell secreting an enzyme or a molecule that will hurt that particular kind of pathogen that's been detected. Now, this might sound like a specific immune response, but remember, this is the innate immune response, and these specific pathogens being detected and these specific responses are extremely general. We're still talking about I've detected a bacteria, don't know what species, but a bacteria. Or, I've been infected by a fungus, don't know what species of fungus, but it's still a fungus. Or, I've been infected by a worm, I don't know what kind of worm, but I got this big thing wriggling around inside of me. It's not a bacteria, it's not a fungus, it's a worm. So we're just talking still about these big, broad categories, and these are all still part of the innate immune response. Okay, so let's come back to the white blood cells. A type of molecule that's secreted by white blood cells are called cytokines. Cytokine molecules are like signals to other immune cells, encouraging them to come en masse to the site of the infection and to respond in a particular way. This release of cytokines and other anti-pathogen chemicals will cause inflammation in the animal's tissues, which is this is the last part of the innate immune defenses before the adaptive immune systems come fully online. So inflammation is really the climax of the innate immune system's response to a pathogen infection. When a pathogen enters the animal's body, it'll encounter a white blood cell called a macrophage. And when the macrophage detects this pathogen, becomes aware of it, it's like, hey, you're not supposed to be here. The macrophage will then release chemokines, which are messenger molecules that recruit other immune cells to come and fight the pathogens. As these chemokines are released from the macrophages, from these white blood cells, immediately after they encounter the pathogen, the chemokines will diffuse through the blood and create a concentration gradient. And an immune cell that's following the increasing concentration of the chemokine will be led to the wound or to the site of the infection. Another part of the inflammation response involves mast cells releasing histamines, which are chemical signals that cause blood vessels to constrict so as to reduce blood flow towards the open wound and thus reduce blood loss. This immediate response to pathogen entry signals the neutrophils, which are another type of white blood cell that come racing to the site of infection, The neutrophils can neutralize the pathogens by consuming them through phagocytosis and then chemically destroying them, breaking them down. As time goes on, more and more immune cells will arrive to the wound or the site of infection, and they will all secrete cytokines that establish the full inflammation response, characterized by the heat and swelling from the sheer accumulation and biochemical activity of all of these immune cells. This state of inflammation will be maintained until the pathogens are cleared out, and there's no more antigen signals propagating the innate immune response. At this point, all that's left is the healing process, which begins pretty much the moment the wound happens with, platelets and clotting and all that good stuff. Alright, so that is the innate immune response in a nutshell, and it exists in virtually all animals. However, for many animals, this is all they have. This inflammation response and the associated cytokine storm, and the very rough mechanism for making broad, very general determinations between the type of pathogen, this is the full extent of most animals' immune capacity. Only the vertebrates have evolved an adaptive immune system that can remember previous infections and make targeted defenses to rapidly destroy the pathogen should it come by again in the future. This adaptive immune system can differentiate not only between fungus and bacteria and worm, these broad categories, but it can also differentiate between genera and species and even subspecies. So how does the adaptive immune system do this? Is there a limit to how many antigens it can detect and make a specific response to? The answer, as far as we can tell, apparently, is no. There is no conceivable limit. And this is because of the molecular nature of antibodies, which are the secret to the adaptive immune system's diversity and specificity of responses. Antibodies are proteins that get secreted by immune cells, and they bind to specific parts of a particular antigen. The interesting detail is that the body does not have individual genes for every single possible antibody. If it did, our genomes would be much larger than they are. But the antibodies are in fact more like custom proteins, made on demand to fit the offending pathogenic antigens. Now, before I get into antibodies, I want to very briefly discuss the immune cells that secrete them. These are the lymphocytes, the white blood cells that live in the lymphoid system and perform the adaptive immune response. There's two types of lymphocytes. You have the T cells and the B cells. Now, both the T cells and the B cells are produced in the bone marrow, but the juvenile T cells will migrate out of the bone marrow through the vasculature to the thymus, where they'll mature and they'll be given the genetic and biochemical instructions, or lessons, I guess, to seek out and destroy the animal cells that are infected with viral particles. Now, the B cells, they're also produced in the bone marrow, but they don't leave. They'll stay and mature in the bone marrow, and then they'll get released into the blood and the lymphoid tissues to detect antigens and produce antibodies. So they serve two different functions here that we can see. The B cells, they produce the antigens and the antibodies, and the T cells, well, they also do a bit of that, but their main function is to destroy the body's own cells that are infected with viral particles. So if your own cell is sick and it's not going to get better and it's a breeding ground for more viruses, the T cells will come in and kill it so that they can sabotage the viral replication and stymie this part of the infection. Alright, now I mentioned the lymphoid tissues. The lymphoid tissues are basically the dedicated tissues of the immune system. They include the lymph nodes, the spleen, and the branching lymph-filled vasculature of the lymphatic system that connects it all together. There's also the mucus-secreting tissues within the orifices, called MALT, or mucosal-associated lymphoid tissue. The MALT lines the points of entry to the body, like the respiratory tract and the gut, and they host populations of white blood cells that act as the first line of adaptive defenses. When a lymphocyte detects an antigen, it migrates to the spleen or into a lymph node where it then becomes activated. To begin the adaptive immune response, an antigen first has to be recognized. This is done by B cells. The B cells have receptors for various antigens called B cell receptors, or BCRs. These BCRs are composed of proteins called chains, And there's light chains, which are pretty small proteins, and there's heavy chains, which are twice as long. And all of these chain proteins are stapled together with disulfide bonds to build the B-cell receptor. There's also a pair of proteins that anchor the B-cell receptor into the cell membrane. And so from this anchoring point, the B-cell receptor extends up. You have two heavy chains extend up, and then they branch outwards, so it forms a Y shape. So the BCR is like a Y shape protruding from the surface of the B-cell. Now the light chains are attached to the lobes of the heavy chains that form the arms of the Y. So the stalk of the Y is just the heavy chains, but the arms of the Y are both the heavy and light chain structure. And at the ends of each arm, where you have this light chain, heavy chain combination, these are where the antigen binding sites exist. So the combined heavy and light chains form like a pair of arms, and the binding site is the hand at the end of the arm that can grab a specific antigen. Now the B cell can also secrete antibodies, which are basically these exact BCRs, but they're just detached from the anchor proteins, keeping them bound to the membrane. So they're the BCRs, but just free-floating in the cytoplasm or in the blood plasma. Now the T cell also has antigen receptors, and these are called T cell receptors, or TCRs, but they have a different shape, and they function a little differently. Where the BCR is Y-shaped, and it can just detect and directly bind to an antigen that it finds out in the wild, the TCR it's not shaped like a Y, it's shaped like a capital I, so it's just a vertical column with an alpha chain and a beta chain. And it has to have the antigen presented to it in a specific manner by another cell in order to be able to even recognize it. So the TCRs aren't going to just be randomly detecting and binding to antigens like the BCRs do. They function differently. This is a key difference that's involved in regulating the T cell response. So all of these cell receptors, be they on B cells or T cells, or whether they're detached and floating around in the blood or the lymph, they're part of a class of molecules called Immunoglobulin Proteins, or Igs. There are different classes of immunoglobulin proteins, like IgGs, which are just your typical secreted antibody, or IgDs, which are your typical cell-bound receptor, but there's also IgEs, which are antibodies secreted in small amounts to fight off parasitic worms. IgAs are pairs of antibodies, or antibody dimers, which are found mostly in malt regions, and keep bacteria from sticking to cell membranes. So it's a way to kind of clean house, sweep the floor, get the bacteria from sticking to the mucous membranes. The IgMs are large groups of antibodies that are like mobile antigen collection platforms that float together through the blood and the lymph. When a BCR or a TCR does bind to an antigen, it binds to a very specific region on the antigen called the epitope. The epitope is basically a point on the molecular structure of the antigen that the antibodies have the ability to recognize, and they can bind to it. So it's kind of like if you had to carry a big heavy object and it's kind of awkward, but there's some convenient handles. Okay, you would hold onto the handles. That's the best place. You have the highest affinity for the handles. If there's no place for the receptor to get a hold onto the antigen, if it can't bind to it, then binding isn't going to occur. It's not going to detect it, and you're not going to have that immune response. So this is actually one way that pathogens have evolved to outmaneuver the immune system. They've evolved to have antigens, to produce antigens that evade the host's immune system. Now this also brings us to another really interesting topic. There's a huge variety of antigens. So how on earth can the adaptive immune system somehow produce BCRs or TCRs capable of getting a foothold or a handhold binding with all of these different kinds of antigens? Like, how does it overcome this huge diversity? The antibodies are able to do this with the variable regions of their light and heavy chain proteins. You see, both the light and the heavy chains are themselves composed of two regions. One is called a constant region that doesn't change that much, and the other is called a variable region. The constant regions, these are constant across pretty much all the light chains and all the heavy chains, their own types of course, but they're constant. They're like the unchanging base that attaches the light chain to the heavy chain. Now the magic happens in the variable region, where the proteins are customized with unique amino acid sequences. It works this way. So the animal has the genes for several dozen protein variants for both its heavy chains and light chains. All of these variants can be combined to potentially create 10 to the 10th to 10 to the 14th different BCRs and virtually the same number of TCRs. Each one of these receptors would possess a slightly different molecular structure, and the different amounts and locations of reactive molecules within this structure would create a very specific chemical landscape, allowing bonds with only a very specific kind of epitope. So it's this customizable quality of the variable region with its 10 to the 10th to its 10 to the 14th different options, different structures and variations, that allows these antibody proteins to be shaped to fit to at least one epitope on virtually every antigen. And keep in mind that antigens can have more than one epitope. There can be more than one region that could potentially be a handhold. So this is how the adaptive immune system is able to recognize such an amazingly wide range of pathogens. And all of this is just explaining how the lymphocytes recognize and bind to the antigen. What comes next, because we're really just at the beginning of the adaptive immune response, is activation. If a lymphocyte matures and it lives in the body for a while, and it never detects the pathogen that it was programmed to detect, it will eventually die, and its parts will be recycled. But when a pathogen is detected, those lymphocytes programmed to respond to that specific pathogen are sent on a war march to destroy it. The explanation for how the body generates and sends out the right lymphocytes with the right antibody programming is called the clonal selection theory, and it works generally like this. So when a lymphocyte is activated upon binding with an antigen, the lymphocyte will divide and make copies of itself. Those clone lymphocytes with a high affinity for the antigen will find more of it, and they'll bind to more of it, and then they'll make more copies of their own. Those cells with a poor affinity for the antigen will generally not find it, and they'll die out. As time goes on, the activated lymphocytes will persist. They won't die off from disuse. In fact, they'll endure. They'll endure long after the infection is over. They act like silent guardians, waiting and watching for any sign of their specific pathogen to respond. Now, this process of activation works differently in B cells and T cells, but in both cases, it's a very highly regulated process. Unleashing these lymphocytes in the body can be dangerous because they're so powerful, so various mechanisms have been evolved to control their release and their activity. At sites of infection, for example, white blood cells called dendritic cells are consuming antigens. Inside their cellular bodies, the antigens are broken down into protein fragments, or the isolated epitopes, which are then given to a major histocompatibility complex, or an MHC. Now, the purpose of this MHC, with all of its component proteins, is to take these antigen fragments and present them properly on the surface of the dendritic cells, so that they can thus be presented to T cells. Among the T cells, there are CD4 plus T cells and CD8 plus T cells. I'll just call them CD4 and CD8 because it's a bit clumsy to say plus all the time. And there are also Class 1 and Class 2 major histocompatibility complexes. So as any immunology student will be able to tell you, Class 2 MHCs present their epitopes to CD4 T cells, and Class 1 MHCs present their epitopes to CD8 T cells. And this is really a kind of unintuitive and confusing way to label things, but hey, this is biology, where photosystem 2 comes first. Anyway, upon activation, the T cells will divide to produce a bunch of clones. And this happens through a process called clonal expansion, which primes the clones to differentiate for their specific role in the adaptive immune response. CD4 T cells will become helper T cells, which go around and help activate other cells. They're helping because they spread the message and they recruit more cells to the fight. The CD8 T cells become cytotoxic T cells, also known as killer T cells. Now these will target and destroy any of the animal's own cells that have become infected, in a scorched earth policy to deny the pathogen any living cells to feed off of or exploit for reproduction or whatever purpose. Now, in the B cells, activation is a lot simpler. B cells can be activated by simply running into an antigen in the bloodstream and binding to it. They don't need a dendritic cell to properly present it to them like a T cell. After the B cells have been activated, they'll mount their newfound antigen onto a class II MHC, and this allows them to be recognized by activated helper T cells. By binding to the class II MHC on the B cell, the helper T cell can then help the B cell reach full activation, and the B cell, once it reaches full activation, will then replicate and create clones of its own. Some of these clones will go on to turn into plasma cells, and these plasma cells are almost ready to start fighting the infecting pathogen. But first, they have to put on their armor and sharpen their swords. To do this, the plasma cells will go to a region of the lymph node, of a local lymph node near the site of infection or wherever these B cells and T cells have been active. They'll go to a local lymph node called a germinal center, where they undergo a process called somatic hypermutation. So here, in the lymph node, the DNA that codes for the variable region of the antibodies will undergo a rapid series of controlled mutations, which then generates a wide diversity of antibodies. These antibodies will have varying affinities for the antigen that's been brought back to base to be researched. Those antibodies that bind strongly to the antigen are preserved, and those that don't, well, they're just left to decay and fall apart and get recycled. When the plasma cells finish this process of somatic hypermutation, the remaining clone lineages will have mutated a viable antibody that has a very high affinity for the antigen. It can strongly bind to the antigen. This is to say that the plasma cells have sharpened their swords, and they're now ready to go out and fight. So they release antibodies, and these antibodies flow to the site of the infection, and because of this somatic hypermutation, they've been tailor-made, they've been precision-carved to find, bind to, and report these antigens. And this is where the adaptive response really begins, where it really gets going. So the adaptive immune response to a pathogen involves a lot of different mechanisms, but these can be organized into two broad groups. On one side, we have cell-mediated responses, so these are responses that the cell is involved with, like local cells or immune cells. And then on the other side, we also have humoral responses. Now in the cell-mediated response, activated CD8 T cells will engage in cell-to-cell scorched earth combat by finding the body's own cells that have been infected and killing them, as a way to starve out or destroy the replicating base of the virus or whatever pathogen. On the other hand, the humoral response involves a much more decentralized mechanism, where instead of individual cells going around killing other cells, you have antibodies and other particles and proteins that have been secreted by other cells. These antibodies and particles and stuff, they're just floating around the blood and the lymph, moving through the vasculature as a kind of environmental variable that can bind to any antigen they find. All of these antibodies flowing to the site of the infection will start binding to all of the pathogens and the antigens that they encounter, and after a while, the pathogen particles will be coated in antibodies. This is a process called opsinization, or if you translate the root words, it means preparation for eating. After the pathogen has been covered in antibodies, they become delicious targets for macrophages and other phagocytes. So these larger immune cells will swoop in, consume the pathogen, and then chemically break it down into smaller fragments. Opsinization is also really neat because the physical coating of antibodies prevents the pathogen from easily binding to stuff, which suppresses the pathogen's ability to infect new cells. So opsinization, the binding of all these antibodies, has two purposes. It slows down the infection until all of the pathogens are neutralized, and it allows the immune cells to eat the pathogen. In addition, some antibodies have more than one binding site, and they can bind to not just a pathogen, but to other antibodies too. You could have multiple pathogens, all coated in antibodies, that can find themselves all getting stuck together in a giant mass of neutralized pathogenic cells. This is called agglutination, because the antibodies virtually glue all of the pathogen cells together. The pathogens can't infect host cells when they're all glued together in a big pile like this, and this big pile is also a super attractive structure for the macrophages. It's like an all-you-can-eat breakfast buffet. Now, if all of this wasn't cool enough, there's also a group of proteins that are a part of what's called the complement system. It's complementary to the main immune system. So, when there's a bunch of pathogenic cells all tied up and glued together with antibodies, these little complement proteins can come by and assemble themselves into complexes on the antibody-covered antigens, and then they literally just blast holes in the plasma membrane of the pathogen cells. Because these pathogenic cells are often neutralized or agglutinated, they're defenseless. So this puncturing attack from the complement system is literally like shooting fish in a barrel. It's going out and executing all your prisoners. It's a pretty ruthless way to wage war, but it's effective. Now, all of these strategies are for destroying extracellular pathogens, like bacteria or small parasitic worms or whatnot. But when the pathogen is intracellular, like a virus, the infected cells will try to fight them off. Some, but not all of the virus particles will get destroyed by the cell fighting back against its own infection, and the viral particle fragments can be presented on the outside of the infected cell. These viral fragments get mounted in the class 1 MHC proteins on the outer surface of the cell, where they're like red flags, big blaring beacons that get recognized by other immune cells, like the T cells. This is like a warning, where the infected cell is saying, "Hey, look everybody, I'm infected. Here's some parts of what's infecting me. Watch out for this." The activated CD8 T cells will then anchor themselves to the infected cell, and they'll release a series of proteins. The first proteins will form pores in the membrane of the infected cell, kind of like a black ops team blasting through a wall to get inside a building. When the pores are made, the CD8 T cell will then secrete apoptosis activating proteins, which flow through the pores into the infected cells. These apoptosis activating proteins will then cause the infected cell to commit apoptosis, to commit suicide. And this is as if the Black Ops team stormed the building and planted a bomb to deny it to the enemy. By destroying the infected cell, the intracellular pathogen is unable to replicate, and the infection is suppressed. Now, as I've talked about the innate and adaptive immune systems, I've compared them. I've talked about the speed of the reaction, about its specificity, and earlier I mentioned the memory. Now, the innate immune system has no memory, but the adaptive immune system does. The adaptive immune system holds grudges. It remembers. The adaptive immune system remembers which pathogens have the audacity to violate the sacred temple that is the animal's body. And the adaptive immune system works to make sure that any future infections, any future blasphemous parasites or pathogens or whatever, will be shut down quickly and without mercy. To establish this immune memory, activated B cells and T cells can reproduce to create a specialized kind of daughter cell called a memory cell. Now, these memory cells are not frontline warriors. They don't participate in the primary immune response, and they don't participate the first time the adaptive immune system runs into a new pathogen. That's not what they're for. This isn't their battle. Instead, these memory cells accumulate in the spleen and the lymph nodes and other bunkers in the lymphatic system, where they can live dormantly for years, if not for decades. Now, if the same pathogen infects the animal body at some point in the future, years or decades in the future, these memory cells can detect the antigens from that pathogen, and they'll initiate not a primary immune response, but a secondary immune response, which is much scarier. The secondary immune response is much faster, it's much more efficient, and it's much more targeted. There's more antibodies that are produced and released to go tag all of the pathogens, and they are laser focused on their target, as the antibodies have been shaped by the somatic hypermutation to have an extremely high affinity. They're very good at finding and binding to, whatever antigen it was that they were designed for. This secondary immune response is much more powerful than the primary response, and it can destroy an invading pathogen before the animal even realizes that it might be sick. That's how powerful it is. Now, this is also the premise for how vaccines work. A vaccine is the injection of an inactivated or a dead pathogen, or a fragment of a pathogen. The immune system will take this inactivated pathogen or a fragment of the pathogen and use it as an antigen. It'll create a primary response, and this will then generate memory cells that will remember that antigen and that pathogen for years. When the vaccinated animal meets a live version of the pathogen out there in the wild, or if you're a vaccinated human and you are exposed to the pathogen that you've already been vaccinated for, you've already been primed for it, so your body initiates a secondary immune response that can easily wipe out the pathogen in a relatively brief period of time. This is the power of the immune system. It's incredible. It's nothing less than an awesomely complex chemical defense grid that evolves in real time to fight off the dynamic pathogen load that the animal is exposed to day to day, year to year, wherever its habitat happens to be. The animal is a chemical superstructure, a massive orchestra of trillions of atoms, and the pathogens want to come in and yell and kick and steal the instruments and wreck it all so they can exploit those chemical orchestras for their own purpose, which is usually to produce more pathogens. But the immune system can put a halt to that, protecting the macroscopic animal from the countless microscopic threats that it faces on a daily basis. All right, wow, this was an intense episode. I had a lot of fun writing it and doing the research, and I hope you found it just as exciting to listen to as I did. When I was an undergrad, I took a mixed undergrad-graduate level immunology course, and it blew my mind on a weekly basis. It is such an amazingly complicated system. You always know at an abstract level that the body is complicated, but if you really want to learn just how insanely complex it is and how all these different things, all these different factors like T cells and B cells and antibodies and maturation in the thymus and hormones and nutrients and blood flow and how all of this stuff is involved in this giant interconnected system, you dive into that through the immune system angle and it's just like, wow. It's mind-bogglingly complicated. It's amazing. And I hope I was able to give you that impression as well. Alright, well, we're at the end of the episode, so it's time to hit that like button if you enjoyed it, and hit subscribe to the channel if you like my content. And if you're a bit iffy on subscribing, well, consider this. In the next episode, moving along in this second series on animal physiology, we'll be discussing my number one favorite animal body system. In the next episode, we'll be discussing the nervous system. I'll be talking about nerves and neurons, synapses and plasticity, the anatomy of the spinal cord, and the various functions and regions of the brain. We'll also talk about the evolution of the brain and the nervous system, what it's for in an evolutionary context, and how evolutionary pressures have shaped the various nervous systems and brains of the animals. So if that sounds cool to you, then hit that subscribe button, and also hit the bell because YouTube can be iffy sometimes. And that way, you'll be sure to see the episode right when I post it. You can check it out as soon as you want. If you want to support the podcast in other ways, then consider hitting up the store or becoming a patron supporter on Patreon. And as always, thanks for listening.